Welcome to the 2010 International Symposium on Dupuytren's disease. We have an incredible lineup of speakers, really an all-star cast representing experts in every field relating to Dupuytren's. I'm pretty excited. I'm Dr. Charles Eaton, call me Charlie. I'm a hand surgeon, and this is why I'm here today. First, a word from our sponsors. I have none. My story starts years ago. Doctors Hurst and Badalamenti up in Stony Brook, New York, started working on a collagenase injection treatment for Dupuytren's. Their industry partner hosted an online patient discussion forum for Dupuytren's. A patient read about doing needle release on that forum and came to my office and asked me to go to Paris and learn it. And I did. Three things happened. First, I became very busy treating Dupuytren's, and at this point I've treated something over 8,000 Dupuytren's hands with this technique. Second, I realized that I'm very lucky, and I would like to give back to the community that gave me this great opportunity. I've met amazing people involved in Dupuytren's, like you. I met Dr. Wolfgang Watt, who set up the Dupuytren Society. He and I actually hatched the idea to launch this symposium. Third, I realize every day that I don't understand Dupuytren's, and it's driving me crazy, and it's a problem which we're all interested in. And any of us could have Dupuytren's. With genetic testing, we could probably demonstrate that I am a distant relative of a number of you in the audience through a common ancestor. And if we had better genetic information on Dupuytren's, we could probably also test whether that common ancestor had the gene for Dupuytren's, because any of us could have it. And the quest to find the best treatment for Dupuytren's has puzzled the smartest people in the world for hundreds of years. In 1974, Marcus Land wrote a piece called Mysterious Aspects of Dupuytren's Contracture. And he asked a few simple questions, such as, if Dupuytren's is not a tumor, if it's not an infection, why does it behave like one? Why is it so unpredictable? If it's a disease of fascia, how do you form cords in areas where there is no fascia? 40 years later, we have only more questions. Could it be a congenital viral infection? When is therapy not enough or too much? If it's genetic, why is it so variable, both individually and between different populations of affected people? But there really is just one question that we should be asking, which is, what do we have to ask? What do we have to know? And what do we have to do to find a cure? Well, we start with meetings like this. This is the 6th, possibly 7th international meeting on Dupuytren's disease that I could track down. The first was in first was in Paris in 1966, hosted by Gossett, Tubiana, and Houston. After the Paris meeting, there was a meeting in Torino, Italy. I couldn't get much information on. Professor Malacy hosted the 1983 Vienna Congress, and then we went across the big pond. McFarlane hosted the 1985 Congress in London, Ontario, and then back to Hanover, Germany in 1991, where Drs. Berger, Brenner, Delbruck, and Hintzman had the 1991 Congress. 20 years passed. Recently, Drs. Hurst and Badalamenti hosted a symposium in Stony Brook, and now in Miami. These meetings have had three main topics: science, surgery, and disease modification. And I went through review books on Dupuytren's to look at what actually was covered. And if you count the number of pages devoted to each topic in review books on Dupuytren's, you'll find that they're mostly split between science and surgery. But that little green margin at the top, disease modification, never gets much coverage. That's what we really need, because Dupuytren's is not a surgical disease. Dupuytren's is a medical condition that does not yet have a good medicine. And we need to work together with that in mind. My hope with this symposium and similar efforts is to foster efforts to fatten up that green line, to push for disease-modifying approaches to Dupuytren's. We'll always need surgery, but we won't always need much. Dupuytrens. Each hand with Dupuytrens is unique, like snowflakes. One process, infinite variety. These are all patients who have Dupuytrens who are waiting for treatment. I'm a hand surgeon. I've done a lot of traditional fasciectomy for Dupuytrens, and every surgeon in this room can tell you that the majority of patients who have surgery for Dupuytrens do pretty well. That's not why we're here. We're here because the same surgeons can tell you that a significant minority of patients who have surgery do very badly. And it is not due to our efforts, it's not due to the surgeon or the surgical technique. These patients don't come back for another surgery. They say things like, if I had known what I was going to get into, I never would have had that surgery. And those are the patients who come to my office every day. Those of you who don't offer alternative treatments for Dupuytren's have no idea, really no idea at all, how many people there are out there who have Dupuytren's, want to be treated, but don't want and will not have surgery. There are a number, really an infinite number of ways to have bad results after surgery for Dupuytren's. And after surgery, Dupuytren's hands are also, they're unique, like snowflakes, except with scars and sometimes fewer fingers. Each of these hands has had surgery for Dupuytren's, and from a distance, it's interesting, they start looking like the hands I showed earlier that were waiting for surgery for Dupuytren's. These hands are trying to tell you something. Each hand tells the story of a real person. This is the maternal grandfather of my 4 boys, who's trying to straighten his fingers after having had 3 operations on each hand. His surgeon at this point had told him, go home, Bill. There's nothing else to do. Now, Bill drank a bit, and he found that, like his alcohol, his Dupuytren's was, it seemed simple, but it wasn't. It was puzzling. In some ways, it was almost impossible to understand. And the more you look at Dupuytren's from any angle, the more impossible it seems. It seems simple, but it's not. And the reason is we don't understand it well enough yet to simplify it. And for that reason, parts of it remain an impossible project. How do you solve an impossible problem? You question what you're doing, you look at how you fail, and you engage outsiders. Now that's the easiest thing to do because we're all outsiders. None of us truly understands Dupuytren's, but we each have the gift of a unique perspective to bring to the table. We need to learn, simplify, and communicate. We're blocked apart by different languages, by different professional environments. We are like a twisted Zen koan. If a tree falls in the woods and someone sees it and writes a book about it, but doesn't write very well, and the book goes out of print and some time passes, then did anyone ever discover a falling tree? The answer is no. My background is initially physics, and even brilliant physicists can drop the ball and fail to pass on information, and this story applies to us. Cavendish was a brilliant chemist. He discovered hydrogen. He discovered that water is a molecule, not an element. He did a lot of work on electricity in a secret laboratory. He wrote his stuff down, but he didn't communicate well because he never published his manuscripts on electricity. And they sat for 100 years before they were picked up by James Maxwell, who read through them. Maxwell went on to develop theories relating electricity and magnetism. Maxwell couldn't simplify. He published his work, but his formulas were so complicated, so confusing, they were useless. And his published works sat basically unused for 50 years before they were picked up by Heaviside. Heaviside could simplify. He spent a few years studying Maxwell's work, and he invented a whole branch of mathematics just to simplify it and came down with what we now know as Maxwell's equations of electromagnetism. But Heaviside didn't communicate his whole life, despite the fact that he invented the technology that gave us long-distance telephone communication. He spent most of his life working on a thing called unified field theory, putting all of physics together. But he never published his manuscripts. Incredibly, he He stuffed his manuscripts under the floorboards of his attic along with other papers to act as insulation in the winter. And they sat there for 40 years, and they weren't discovered until 2 years after the death of Einstein. Now, that's a real shame because, Einstein published E = MC squared as a young man, but he had spent most of his life working on unified field theory. Now, imagine how differently things might have turned out if Einstein had had access to Heaviside's work. But we'll never know. Just as we'll never know what Einstein's last words were, because the nurse taking care of him in his final days didn't speak German. She couldn't understand what he was saying. What a waste. We cannot work in isolation. We have to figure out better ways to share what we know, to put ourselves together. Even my computer knows this. I was testing out a voice transcription system last week, and I said, Dupuytrens, and the computer typed 2 bedrooms. 2 bedrooms. And I thought, it's incredible. Even my computer knows that we can't stay in separate bedrooms if the goal is to give birth to something new. McFarlane found the same thing. Preparing for the Ontario Congress, he went through libraries around the world. He hired linguists and translators. He wanted to put all the scientific records together. And what he found, to his horror, was again and again, the ball was dropped. Things were written down and lost in print. And we cannot afford to do the same thing. Part of the problem is that Dupuytren's is several things all at once. It's genetic, it's mechanical, it's biological. You can't separate these and think that you're going to understand Dupuytren's. Fasciotomy, fasciectomy, collagenase injection, cell biology, molecular biology. You can't separate these things if you really want to understand Dupuytren's. We have to communicate. We are not all that different. Like this surgeon and 2 biologists discussing Dupuytren's. We may seem different, but we're not. Well, maybe it's a biologist and 2 surgeons. The biology of Dupuytren's may seem complicated, but it's because we don't understand it well enough to simplify it. We are like the story of the blind man trying to describe an elephant. Describing something is not the same as understanding it. Now, I'm a surgeon. I look at diagrams like this, and all the data's there, but I can't understand it. I don't know those abbreviations, and even if I did, to me, it looks like this. Now, is TGF-beta the white car? I can't read the license plate. This is more my speed, cartoons. The point is that you have to, unless you can simplify something enough to explain it to someone else and have them understand it, then you don't understand it. And you have to be careful about simplifying. Simplify too much, lose the point, and then you don't know where to start. Now, I can understand how unpredictable is the outcome of a process that has many steps toward its end. But even this has to be simplified down to the most essential steps to be able to quench my thirst for knowledge. That's not the only problem. The second hurdle that we have to overcome is the difference between our understanding and reality. The gap between the laboratory model and the real thing is just as big a hurdle as the gap between the clinical plan and its execution. And we have to get past this. And we can. Why? We have 4 great resources. The first is us. We are all explorers. We have a need to discover, everyone in this room. And we each are comfortable with difficult challenges, each in our own environment. Because we are who we are, we've each seen things in our lives that few other people in the world have seen. And that gives us unique perspectives to contribute together as a group. And because of that, we also know that unless we reach into the unknown, we'll never know what lies within our grasp. Our second strength is that we are all lifetime students. We are academics. Our lives are about learning. We appreciate the beauty of science in the universe like this, 150 light years away, the Hand of God nebula. And we appreciate the beauty of mathematics in everyday things. We have 3 very interested parties who can and must work together because they're all interested in the same thing. That's our 4th resource. And our last resource is that we have the power of the internet giving us tools that we didn't have at the time of the last European Symposium. Dr. Watt created the Dupuytren Society, harnessing the power of patients online. I set up the Dupuytren Foundation to try to fund research to find better treatment options together. And the point of this symposium is to create an academic international task force of people who are interested in creating and carrying out large-scale, multi-center collaborative studies to find better treatment options, to work for a cure. Up until this symposium on Dupuytren's have been about state-of-the-art, the present. This symposium is about the future. Inroads into disease modification for Dupuytren's has trickle-down effect for other fibrotic conditions, pulmonary fibrosis, scleroderma, renal interstitial fibrosis, cirrhosis. We stand on a great threshold because Dupuytren's is the flagship fibrotic condition. So, welcome to the 2010 International Symposium on Dupuytren's Disease.